Hi everybody, I hope you're well and welcome to our second unit. In this unit we are going to be focusing on RNA, which stands for ribonucleic acid, and just like DNA, RNA is an example of a nucleic acid. However, there are some differences between RNA and DNA, and that's where we will start in this topic. One important difference between a DNA molecule and an RNA molecule is the shape of the molecule itself. In DNA, your molecule is always double-stranded, whereas in RNA, your molecule is generally single-stranded, but it can contain double-stranded regions because it may fold upon itself or twist upon itself. So DNA is double-stranded, whereas RNA can be double-stranded. A second difference is your pentose sugar. Pentose means 5, so we have a 5-carbon sugar in your DNA nucleotides and your RNA nucleotides. However, in DNA it is called deoxyribose sugar because there is one less oxygen, and in RNA we call the sugar a ribose sugar. And the last point to talk about is your bases that we find. The word base here is referring to a nitrogenous base. In DNA we have thymine, whereas in RNA we have uracil. So uracil replaces thymine in an RNA molecule. We will not find uracil in DNA, and we will not find thymine in RNA. So unlike a DNA molecule, in which we just have one type, RNA can exist as many types. There is mRNA, which stands for messenger RNA. There is tRNA, which stands for transfer RNA. And there is rRNA, which stands for ribosomal RNA. And all of these RNA molecules play a very important role in the process of protein synthesis. But before we can jump into their specific roles, let us touch on a few important aspects related to an mRNA and a tRNA molecule. If we had to look at an mRNA molecule, we can see from this diagram that it is indeed a single-stranded molecule, and the nitrogenous bases within an mRNA molecule are arranged in groups of 3. And an arrangement of this or these 3 nitrogenous bases on an mRNA molecule has a special name, and that name is called a codon. When we look at a tRNA molecule, we can see from a 3-dimensional view that this molecule contains many twists and many folds, and it's very complicated to draw. And generally, in tests or exams or studying, we represent a tRNA molecule with this structure that I've just circled in yellow to make it easier instead of drawing out this entire folded and twisted structure. And just like with a mRNA molecule, a tRNA molecule has 3 nitrogenous bases, which I'm highlighting in purple at the bottom, and they have a special name called an anticodon, which we can see in all our diagrams on this slide. And a certain anticodon is complementary to a codon. And a last point I wish to emphasize with regards to a tRNA molecule is that a tRNA molecule carries an amino acid, which is generally found around the top region of the molecule. And an amino acid is the monomer or the building block that is needed to produce a protein. And the production of a protein is largely accomplished by the work of RNA molecules. So from our work on DNA, we saw that a DNA molecule may consist of coding segments and may also consist of non-coding segments. Those coding segments have the code or the recipe that is needed to produce a protein. However, DNA is an extremely important molecule and we don't want it to leave the nucleus because it might get damaged. And that is where your RNA molecules, mRNA, tRNA, and rRNA come into play. So mRNA, tRNA, and rRNA essentially are a middleman between DNA, which has the code to make a protein, and the protein itself. And this process of moving from a DNA to a protein using RNA molecules is called protein synthesis. The word synthesis means to make, so the making of proteins, and it consists of two stages. The first stage is called transcription, which takes place in the nucleus of your cell. And the second stage is called translation, which takes place in your cytoplasm at a ribosome. Furthermore, transcription consists of three phases: initiation, elongation, and termination. And translation also consists of three phases: initiation, elongation, and termination. And at the end, we will have produced a protein, and that is what we want in protein synthesis, to create a protein which is based on the recipe for that protein held on your DNA strand. And next we are going to look at the process of transcription and translation in detail. So we'll start off by looking at the process of transcription. Transcription is the first stage of protein synthesis. And like I mentioned, it consists of three phases, and the first phase is called initiation. So if we have a look at the first diagram on our slide, we can see that we have a DNA molecule. So since we have a DNA molecule, we still are in the nucleus of our cell. An enzyme by the name of RNA polymerase, and by the way, if a word ends in ASE in biology, that often means it's an enzyme. RNA polymerase attaches onto the promoter of your DNA, and your promoter is the start of your transcription unit. And a transcription unit is basically a coding gene. As we mentioned in our unit on DNA, a gene is a recipe for a protein. If we now have a look at the second diagram, we can see that the region in which your RNA polymerase is found, so this is your RNA polymerase in your first diagram, same thing in your second diagram. However, in the region where your RNA polymerase is found, your DNA becomes unwound. And that is due to the activity of another enzyme, which is not labeled on this diagram, but that enzyme is called helicase, which simply weakens the hydrogen bonding between your two DNA strands. We then have two strands of DNA in that bubble, the top strand in red and the bottom strand, which I'll show in light blue. And the strand in light blue becomes what is known as the template strand of DNA. And the word template there means that it is this strand that we are going to use as a template for the process of transcription to take place. We can see that an RNA transcript is starting to be formed, but that we will focus on in our second phase of transcription. Our next phase, or our next part of transcription, is called elongation. It's the second part of transcription. And let's have a look at our RNA polymerase. RNA polymerase is found in this location. However, when we started off, it was found over here at the promoter. So we can see that in our first diagram, your RNA polymerase is moving down your DNA strand along your template strand. And as it moves down the template strand, the RNA polymerase adds in free RNA nucleotides, which are complementary to the template strand DNA nucleotides. So to understand that, let's have a look at our second diagram. So in yellow, I will draw in our template strand, and our template strand reads as T, A, G, G, T, T, A, C, C in this instance. We also have our RNA polymerase, which I will highlight in purple. And as RNA polymerase moves downwards and along your template strand, it adds in free RNA nucleotides to the template strand of DNA, to the yellow strand of DNA. So firstly, a cytosine was added to a guanine, which we can't see on this diagram, but we know it must be a guanine on your DNA template strand. Next, an adenine was added by RNA polymerase to the thymine on your template strand. Next, uracil was added in by RNA polymerase to the adenine on your template strand. Next, cytosine was added by RNA polymerase to the guanine on your template strand, and so on, and so on, and so on. And what we are doing is we are simply, for lack of a better word, transcribing or rewriting this gene in an RNA format. And that RNA format is in fact a pre-mRNA molecule. So in this diagram, they refer to the RNA simply as RNA. However, it is in fact a pre-mRNA molecule. And remember, I mentioned that your molecule, or rather that your RNA polymerase, is moving downwards and it is performing transcription. Once a unit has already been transcribed, the unit will then form a stronger hydrogen bonding, so the effect of helicase no longer plays a role, and it will rewind. and that is the process of elongation, which continues until a long strand of pre-mRNA has been formed. We are still in the nucleus of our cell. We then come to our last phase of transcription, which is called termination. And termination means to stop. So we have got to now stop the process of transcription from taking place. So we can see in this diagram that our entire pre-mRNA molecule has been transcribed, and as a result of that, your DNA molecule has gone back to its original structure. There's no longer any bubbles in that DNA, and the functioning of RNA polymerase is no longer taking place. And so RNA polymerase will leave your DNA strand, and your pre-mRNA molecule, this molecule here, will now leave your nucleus and will then be modified. It undergoes certain modifications on the 5' end and certain modifications on the 3' end. And that will then convert your pre-mRNA into a full-on mRNA. And we have now completed the process of termination under transcription, and termination means that transcription has ended. And at the end of transcription, we have an mRNA molecule, which is simply a single-stranded molecule which has an RNA code of the gene on your DNA template. The whole point of protein synthesis is to produce a protein, and we have not yet produced a protein. And so we now can enter into our next phase of protein synthesis, or our next stage of protein synthesis, called translation, to understand how a protein or a polypeptide can form. So the process of transcription has taken place, and at the end of transcription we formed a mRNA molecule which was modified. And the reason why those modifications happened, these are structural modifications, by the way, the reason why they happened was to ensure that your mRNA molecule could be protected from enzymes in the cytoplasm. Also, it allowed for your mRNA molecule to move freely through the cytoplasm, and it also allows the mRNA molecule to bind to a ribosomal unit. And the moment that your mRNA molecule binds to a ribosomal unit, then we can say that translation can take place. And translation, just as with transcription, is divided up into 3 phases. And the first one is called initiation. So initiation starts when an mRNA molecule, which is shown in red in this image, bonds to a small ribosomal subunit at a region called the mRNA binding site. And once that happens, your small ribosomal unit will move along your mRNA molecule until it finds the start codon. And a codon is a set of 3 nitrogenous bases, and so your start codon is a set of 3 nitrogenous bases which can start the production of a polypeptide chain. If we have a look at this image here, which has been enlarged for us up here, we can see that your start codon has the following sequence: A, U, G. When I spoke about mRNA and tRNA, I said that codons have a complementary anticodon. and an anticodon is found in your tRNA molecules. And so we can now talk about an initiator or a start tRNA. The moment that your small ribosomal unit moves along the mRNA and finds the start codon, your tRNA molecule, which holds an amino acid, will move in from the cytoplasm and temporarily join to your codon. And if we have a look back at this image, we can see that your anticodon reads UAC, and if we look at the relationship between your codon and your anticodon, we can say that they are complementary because the U on your anticodon is complementary to the A on your codon. The A on your anticodon is complementary to the U on your codon, and the C on your anticodon is complementary to the G on your codon. Specifically, with an initiator tRNA or a start tRNA, it always carries along with it an amino acid called methionine. And this amino acid always forms the start of a polypeptide chain. So once all of this has taken place, a larger ribosomal unit is then added on, and this forms the translation initiation complex, which contains 3 different sites: the A site, the P site, and the E site. And we will talk about more of the aspects related to these sites in the next phase of translation, which is called elongation. So next we will look at the process of elongation as applied to the second stage of protein synthesis, which is called translation. And what happens during elongation is simply the movement of tRNA molecules from the A site to the P site and then to the A site. And as this process takes place, an amino acid chain or a polypeptide will start to be formed. So in order to understand this process, let's call this tRNA 1 and this can be tRNA 2. So remember that a tRNA molecule always holds an amino acid at its end. So in diagram number 1, or let me rather call it diagram number A, we can see that your tRNA molecule number 1 is in the P site, and there is a polypeptide chain which is starting to form. However, your entire mRNA code has not been fully read by your ribosomal unit as yet. In other words, there's still more amino acids which need to be added on to your polypeptide chain. So in diagram number B, we see that your second tRNA molecule has now entered into your A site. And once it's entered into your A site, it will form a bond, and by it I mean the amino acid attached to the end of that tRNA molecule will form a bond to the growing polypeptide chain on tRNA molecule number 1. And if we look at diagram number C, as a result of that, your polypeptide chain will then move over to your second, or rather tRNA molecule number 2. And furthermore, your ribosomal unit will continue to move, reading the mRNA strand. So in other words, your ribosomal unit is still going to move in this direction, And as it moves in that direction, we find that tRNA molecule 1 enters into the E site, and tRNA molecule number 2 enters into the P site. And I like to remember it as follows. The A site, I think of the letter A as being the start of your alphabet. So that's where your tRNA molecules start. Your P site, I think of P as in polypeptide, is generally where the polypeptide formation takes place. And E stands for exit, and that is where your tRNA molecule leaves. So remember that your tRNA molecule does not permanently bond to your mRNA molecule. It's a temporary bond, and the amino acid that is attached to it is left at the ribosomal unit. And so when the tRNA molecule leaves, it attaches to another amino acid in the cytoplasm and will then return back to the ribosomal unit if it needs to return back. And that brings the process of elongation with regards to translation to an end. However, we need to stop the process of translation, and that is where our next phase of translation comes into play. So now we've entered our last phase of translation, which is called termination. And the word termination means stop. So we have to bring the process of translation to a stop. And the process of termination begins when a stop codon reaches the A site within your ribosomal unit. And so instead of a tRNA molecule attaching to the A site, a protein called a release factor will attach onto your A site. And as a result of that protein called the release factor being found in your A site, it will cause a water molecule to be added on to the end of your polypeptide chain. And the moment that takes place, your entire translation initiation complex comes apart. So your smaller ribosomal unit and larger ribosomal unit move away from each other. And when that happens, your mRNA molecule will move away, and we then have a free polypeptide, which is a sequence of amino acids bonded together by peptide bonds. And here we have now formed a protein. So there is a difference between a polypeptide and a protein, but I will just refer to it as a protein for the sake of keeping things simple. So we've now formed a protein, and we've reached our product. The whole idea of protein synthesis was to create a protein. Now that the protein has been created, it will move to organelles like the endoplasmic reticulum and the Golgi body or the Golgi apparatus, where it will then be modified. And once it's been modified, that protein can be used by the cell or transported away from the cell to where it is needed. And that brings the whole process of protein synthesis to an end. So protein synthesis consists of two major phases, translation and transcription. Transcription occurs first, and the product of transcription is an mRNA molecule, and this process of transcription takes place in the nucleus. Translation takes place after transcription. It takes place at the ribosome in the cytoplasm, and the product that is formed is the product of protein synthesis, which is your protein or your polypeptide chain. So this diagram shows us a summarized, very skeletal understanding of protein synthesis, in which we have your DNA template strand, let me use a darker color, your DNA template strand, forming mRNA through the process of transcription. Your mRNA has codons, and each codon on your mRNA strand has a complementary anticodon, which is found on your tRNA molecules. Your tRNA molecule has an amino acid attached to it, and through the process of translation, we can link up those amino acids via peptide bonds to form a polypeptide sequence, which can undergo modification and become a protein. But the key idea behind this flowchart is showing us that when we know one bit of information, we can determine the rest. So for example, if I've been given a DNA template sequence or a DNA template strand, I can work to get the mRNA strand, the tRNA strand, and the amino acid sequence by remembering the complementary idea. Or if I have an amino acid sequence, I can work backwards and get the mRNA strand and the DNA template strand, which codes for that amino acid sequence. The table shown on this slide helps us to determine the amino acid sequences that are coded for by codons on your mRNA strand. So the key idea when answering questions relating to cracking the code is remembering the complementary idea. Remember that your DNA template strand is complementary to your mRNA codons, and your mRNA codons are complementary to your tRNA anticodons. So that brings our unit to an end. I hope that you enjoyed this video, and I hope that protein synthesis makes a bit more sense. Thank you for watching, and I'll see you soon.